Hello everyone, welcome to a study of the points of Section 4, Biological and Clinical Importance of Epigenetics, Part 2. Covalent modifications of histones can regulate all DNA-dependent processes. It has become more and more evident that histone modifications are key players in the regulation of chromatin states and dymetics as well as in gene expression. Therefore, histone modifications and the enzymatic machineries that sit them are critical regulators that can control cellular proliferation, differentiation, plasticity, and malignancy processes. Histone acetylation by HAT can alter closed chromatin by relaxed chromatin, while HDAC catalyzed deacylation has the opposite function. This can affect gene transcription and thus can change biological efforts in malignant cells, including cell proliferation, cell cycle, migration, and cell death. Besides transcription, new roles for histone acetylation have been uncovered and include nucleosome assembly, chromatin folding, heterochromatic silencing, DNA damage repair, and replication. Tumor suppressor genes in normal cells are involved in HAT-associated acetylation in co-activator complex, while in cancer cells, tumor suppressor genes are in co-repressor complex, which include HDAC for deacylation. So, the special HDAC inhibitors can prevent the deacylation, affect gene expression, and be used for tumors treatment. The inhibitors can control protein stability by induction of oncogene-expressed protein degradation and inhibition of tumor suppressor degradation. They can induce cell cycle arrest by induction of cell cycle inhibitors and inhibition of cell cycle progression-related genes, inhibition of telomerase activity, induction of differentiation, and apoptosis are also involved in the functions of HDAC inhibitors. Histone deacetylase inhibitors are used to treat cutaneous T-cell lymphoma. Drugs include Atodax, Belodacul, Faridacar, and Zoaneza. Methylation of histone tails occurs mainly at lysine and arginine residues. In contrast to acetylation, addition of a methyl group on a side chain doesn't alter its charge, and therefore it is unlikely that methylation would directly modulate nucleosomal interactions required for chromatin folding. Another distinct characteristic of histone methylation is that it has been implicated in both transcriptional activation and repression. Methylation may regulate transcription and enable gene attendance in an on or off state. Methylation has been considered to be an essential step involved in many cell fate determinations, such as developmental and differentiation processes, pluripotency, and maintaining genomic integrity. It is highly associated with cancer and other diseases, and approximately 5% of HMTs encoded by the human genome are now linked to diseases and in particular tumorigenesis. Doubt, genetic alterations, and tumor microenvironment can affect cell metabolism and thus regulate histone methylation and demethylation by HMT and HDM respectively. This can cause an activation and repression of genes expression for tumorigenesis, dedifferentiation, and drug resistance. Perspectives for targeting histone methylation by HMT inhibitors or HDM inhibitors for cancer therapy have been in preclinical process. Histone ubiquitination refers to the transport of ubiquitin to the histone core proteins such as H2A and H2B. H2A ubiquitination through PRC1 typically represses gene expression, while H2B ubiquitination can both activate and inhibit targeted gene expression. In histone modification, Enzyme 1, Enzyme 2, and Enzyme 3 can catalyze the ubiquitination of histone-modifying enzymes step by step for their degradation. On the contrary, DUBs can proceed deubiquitination for the substitution of histone-modifying enzymes. So, this regulation can also be involved in transcriptional elongation, DNA damage repair, cell proliferation, tumorigenesis, metastasis, stem cell renewal, and etc. SUMOR is the abbreviation of small ubiquitin-related modifier. SUMORylation, which is also included in histone modification, is long thought not to target proteins for degradation. But accumulating evidence suggests that it can serve as a primary prerequisite for ubiquitination, thereby inducing protein ubiquitination and degradation. For example, H4 is modified by SUMOR family proteins, which binds to the SUMOR conjugating enzyme, that is enzyme 2 and UBC9. Then H4 can be simulated in enzyme 1, that is SUMOR activating enzyme, and enzyme 2 dependent manner. Thus, there is an important crosstalk between SUMORylation and ubiquitination in determining protein fate. Deregulation in these two processes may cause aberrant activity of proteins and in turn contributes to cancer development. MicroRNAs are defined as 21 to 25 nucleotide single-stranded RNAs without protein products, which are produced from helping-shaped processors. In nucleus, genes for microRNAs are transcribed to a primary microRNA, prime microRNA. The prime microRNA is processed within the nucleus to a processor microRNA, pre-microRNA by DROSA, a class II RNase 3 enzyme. Next, the transport of pre-microRNAs to the cytoplasm is mediated by exportin 5. In the cytoplasm, they are further processed to become mature microRNAs by Dicer, an RNase 3 type protein, and loaded onto the arginate protein to produce the effector RNA-induced silencing complex RISC. The majority of microRNAs are located in intergenetic regions, whereas a few are annotated in intronic regions. MicroRNAs guide microRISC to specifically recognize messenger RNA, mRNA, and downregulate gene expression by post-transcriptional mechanisms, translational repression, mRNA destabilization, and mRNA degradation. Studies indicate that most microRNA binding sites in mRNAs lie in the 3' UTR as multiple copies. MicroRNAs bind with mismatches and biages through Watson-Crick base pairing. The degree of microRNA-mRNA complementarity is a major determinant of the regulatory mechanism process. The high degree of complementarity enables the agile catalyzed degradation of target mRNA sequences through the mRNA cleavage mechanism process. In contrast, a central mismatch omits degradation and facilitates the translational repression mechanisms. One microRNA can actively regulate tens to hundreds target gene expressions, and its functions are oppositely determined by the functions of the targets. So, the microRNAs that target oncogenes, mRNA, and proteins are classified as tumor suppressors, while the ones targeting tumor suppressors are classed as oncogenes. mRNAs can also be involved in different regulatory mechanisms and signal pathways. For example, the microRNA LAT7 can inhibit cell cycle, cell growth, and migration by negatively regulating the corresponding targets. MYC and NF-kappa B can regulate the level of LAT7 expression by Lin28. The misregulation of microRNAs is linked to the development of certain diseases. It has been demonstrated that the restoration of misregulated microRNAs to their normal levels can reduce or even eliminate diseases including tumors. Because microRNAs are naturally occurring molecules, there are certain advantages in their application as therapeutic agents. Worldwide researchers have validated the theory of microRNA replacement therapy, which involves introducing synthetic microRNAs or microRNA mimickers into diseased tissues in an attempt to restore normal proliferation, apoptosis, cell cycle, and other cellular functions that have been affected by the misregulation of one or more microRNAs. In contrast, some researchers have artisanized microRNA inhibitors in an effort to increase the angiogenesis levels of therapeutic proteins. In many cases, the reactivation or inhibition of these microRNA-regulated pathways leads to a significant therapeutic response. The pioneering companies have initiated studies on creating valuable therapeutic candidates with microRNA inhibitors and microRNA mimickers in diverse fields such as cancer, cardiovascular diseases, neurological disorders, and viral infections. So it is basic to get the hallmarks of the diseases, for example, breast cancer, and thus to find the corresponding microRNA biomarkers for diagnosis and treatment. For example, the MicroView kit launched by Rostic Genomics uses the microRNA expression characteristics of lung cancer, which can differentiate the 4 main subtypes of lung cancer by a small amount of microRNA detection of cancer cells through patient puncture. In conclusion, both endogenous agents and exogenous agents can cause DNA damage, including epigenetic alteration and somic mutation. If DNA repair is deficient, it is suggested large increase in epigenetic alteration and somic mutations, which may result in progression to cancer. The available data indicate the great potential of epigenetic drugs in cancer therapy. That's all for this section. Thank you.